Hello everyone, my name is Arvind and the title of my talk is Patient-Derived Xenograft-Based Pharmacogenomics for Precision Medicine. Cancer is one of the leading causes of death worldwide. In cancer, the treatment efficacy is low. In last few decades, we have made tremendous progress in sequencing technology. And this has made it clear that cancer is a complex genomic disease. Therefore, the traditional approach of having a single drug for all cancer patients do not work in cancer. Precision medicine aims to change this situation by matching a patient to a drug based on tumor's genomic profile. On paper, the idea of precision medicine is great. However, the biggest challenge in precision medicine is to find biomarkers which can predict drug response. As we have only few biomarkers, the matching rate is low. For example, these are some of the major precision medicine clinical trials. Here, the red bars represent percentage of patients for whom we could not find any actionable mutation. The challenge is to reduce these red bars by finding biomarkers for these patients. There are four different kinds of model systems that we use for biomarker discovery. Cell lines are workhorse of the cancer research. They are easy to grow and high throughput drug screening is cheap. However, they are in vitro model and they do not recapitulate tumor biology very well. Organoids are becoming popular. They are easy and is comparatively easy to do high throughput screening. However, they are difficult to grow and most of the time we need a magic formula to grow them. PDXs are in vivo tumor models. It is well-established method and they are known to recapitulate tumor biology very well. Patients are, as a model, are the gold standard. However, there are a lot of ethical issues. Running clinical trial is very expensive also. Given all these advantages and disadvantages of the model systems, there is a push in cancer research to grow more and use more and more PDX models. For example, one of the recommendation of Cancer Moonshot panel is to develop 3D models. So how does the PDX-based pharmacogenomics work? We take a patient tumor sample and we implant it in an immune deficient mice where it grows. When the tumor volume is large enough, we sacrifice the mice and remove the tumor, chop it into pieces and implant it into the new mice. When we want to test a drug, let's say we have 6 PDXs from a patient, we declare 3 as a control and rest we give the drug. We monitor the tumor volume across time. In best case, we are hoping to see something like this where with the time, in the control group, the tumor volume is increasing and in the treatment group, the tumor volume is shrinking. In PDX pharmacogenomics, our aim is to link the genomic data from the tumor to the drug response data from the PDXs to find biomarkers. To manage and analyze this data, we have recently developed our software called Jiva. This work has been published in Cancer Research and package is available in GitHub as well as in Bioconductor repository. To give you an overview what we can do this with this package, we take all kind of genomic data and process it in a unified manner. Then we link the data with the time versus tumor volume data from the PDX experiments. And we create an R object called Jiva set. Once we have this Jiva set, we can visualize this data, we can compute response, we can do biomarker discovery and perform machine learning on this data. We also have functions to directly get the data from Google Cloud or from the Excel sheets as well as from the study log. So here are some of the examples that you can do with the package. For example, you can visualize the PDX growth curves. Here, each dotted line represents an individual PDX and the solid line represents their average. You can change this visualization. For example, you can do the normalized PDX curves and you can compute the error bars and do different kind of ribbon visualization just by simple one-line commands. Another thing we have implemented in our package is different kind of response matrices. As in the PDX field, there is still not a unified response matrix that everybody is using. So we have applied different response metrics such as area between the curve, angle between treatment and growth curve, and so forth. So one of this response metrics is mREGIST, which is a modified criteria of the REGIST criteria that are used in the clinic. So once we have the package, we formatted Novartis PDX data into our package. In 2015, Novartis released a big PDX study containing more than 1000 PDXs. So here you see a visualization of the Novartis PDX data in case of breast cancer using our package. So in this plot, each column represents PDX from a single patient. The control or untreated PDXs are on the first row, and all different drugs that have been tested on these PDXs are shown here. As I mentioned earlier, response is measured using mREGIST, where the blue means we get a response and red means we do not see any response, or we do not get any response. You can create such a plot using just 2 lines of command from our package. In this visualization, you will notice that for certain patient PDXs are not responding to any of the drug, while for certain other cases, the patient PDXs are responding to a lot of different drugs. Such visualization gives you an overview of PDX clinical trial. To use the Jiva package, you will require some knowledge of programming. However, for clinicians and experimental biologists, we have developed a web version of our tool. It's called JivaDB. So if you go to this website, you can, for example, select a data set, and you can select the drug that you want to visualize for the data. And you can also select the gene of interest, and it will show you the drug response as well as corresponding genomic information. You can highlight a particular patient just by clicking on it, and if you click on the cell, you will see the tumor growth curve, PDX tumor growth curve and all different kind of response matrices from this PDX tumor growth curve. Earlier, when I was talking about how we create PDXs, I mentioned the process of passaging. That is taking out tumor from one mice and implanting it in the new mice. In PDXs, we do this multiple times. An important question here is that if the genomic properties of tumor are stable from passage to passage. So we assess this stability of the gene expression across passages by computing interclass correlation for each gene across passages. High ICC value indicate that the expression of the gene is stable across passages. As you can see, most of the genes ICC value is high, so they're stable across passages. However, for certain genes, the ICC value is low, which indicates that expression of these genes are not stable across passages. We also analyze the pathway associated with these genes, and most of the pathways are highly stable across passages. However, we found that there are few pathways that are not stable across passages. So if you're targeting these particular pathways in the PDX experiment, you have to be careful about the passage effect. We also perform the biomarker discovery. So here you see in the left side, you see the gene expression data for the breast cancer patient. And in the right side, you see the association between the gene expression and response of the drug. Here, the size of the dot represents the strength of the association. So to highlight an example, we have shown that ERBB2 gene expression is highly associated with trastuzumab drug response. We also looked at the pathways which are associated with the drug response. Here, we divide the drug into different classes according to their targets. For example, all the drugs targeting MAPK genes are grouped into one class. In the right side, you will see pathways that are significantly associated with drug response. Again, to highlight some of the examples, we have shown that EGFR class of drugs are significantly associated with signaling by EGFR, and MAPK class of the drugs are significantly associated with MAPK activation pathways. So to summarize, we have shown that Jiva provides a novel platform for PDX pharmacogenomic data analysis. Using PDXs, we can predict biomarkers for drug response. Gene expression of the PDXs is consistent across passages, and our Jiva software facilitates biomarker discovery. At the last, I would like to acknowledge all the lab members as well as all our collaborators. Thank you very much.